You know those scientific debates that just never seem to end? Where you've got this one simple question, but for decades, all you get are contradictory answers? Well, that is exactly what we are jumping into today. We're diving deep into the world of animal nutrition to solve a really long-standing mystery. It's about two super important supplements, DL-methionine and its cousin, HMTBA. Getting this right matters. I mean, the health of millions of animals depends on it. So why on earth can't scientists just agree on which one works best? So here's the setup. We've got two molecules, both designed to do the exact same job, and yet study after study comes to a totally different conclusion. It's kind of baffling, right? Is one just secretly better than the other? Is all the science just flawed? Or is there something else going on that we're missing? Well, today, we're going to put on our detective hats, we're going to sift through all the evidence and figure out exactly why good science has been giving us such conflicting answers. Let's start following the clues. All right, every investigation has to start with the suspects, and right off the bat, we find our first huge clue. Even though these two molecules do the same job, we are absolutely not comparing apples to apples here. We are quite literally comparing apples and oranges. And this is where the plot really starts to thicken. So on one hand, we've got DL-methionine. Think of this as our apple. It's a classic amino acid, one of the basic building blocks of protein. But then you have HMTBA, or orange, and get this, it's not an amino acid at all. It's an organic acid. Chemically, it's actually got more in common with the lactic acid your muscles make when you work out. This difference, which seems kind of small at first, is actually the key that's going to unlock this whole mystery. Okay, so if our two molecules are chemically different, it kind of makes sense they wouldn't behave the same way inside an animal's body, right? So let's follow them. Let's trace their journey through the digestive system and see exactly where their paths split. So DL-methionine, our amino acid, it takes the exclusive route. It uses something called active transport. Now, the best way to think of this is like a VIP taxi service for molecules. It needs a special protein shuttle to get across the intestinal wall. And that taxi ride, it isn't free. It actually costs the cell precious energy to run. Super selective for sure, but that exclusivity comes at a cost. Meanwhile, HMTBA, our organic acid, takes a much more direct route. Passive diffusion. Think of this like an open gate at a stadium after a game. There's no bouncer, no special ticket, no velvet rope. It's just a simple flow from where there's a lot of it to where there's less of it. And what's the big takeaway here? No special shuttles, and this is crucial, it requires zero energy. This table just lays it all out so clearly. Look at that top row. One needs energy, the other is totally free. Now look at the second row. This is wild. They aren't even absorbed in the same place. DL-Met needs its taxi service down in the small intestine, but HMTBA, it just walks through the open gateway way earlier, in the acidic parts of the upper gut. We're talking about two completely different journeys happening inside the body. This is a massive piece of the puzzle. In fact, get this. Around 85% of HMTBA gets absorbed before it even gets to the small intestine, which is the main spot for amino acid absorption. Just think about that for a second. That means the job is basically done before the molecule even arrives at the location where most researchers were probably looking for it. This is a huge clue that's been hiding in plain sight all along. So we have two different molecules. They take two completely different paths to get absorbed, and that right there is how we solve this whole thing. The decades of confusing, conflicting research, it wasn't because the molecules were inconsistent. Nope, it's because scientists were often using the wrong tools to measure them. You want to know how to design an experiment that's practically guaranteed to give you a biased result? It's simple. First, use a lab setup that can only see those VIP taxis and is completely blind to the open gates. Well, of course DL-Met is going to look like a superstar then. Then, fast the animal beforehand. That's like putting a bunch of extra taxis on the road just for DL-Met, which skews the results even more. And finally, imagine you're trying to track a package, but your tracking sticker keeps falling off and sticking to random mail trucks. That's what happens with certain chemical tracers. You end up with junk data. A lot of those famous conflicting studies were, well, just using the wrong tools for the job. Okay, so case closed, right? Not so fast. The plot actually thickens a little bit because critics of HMTBA have another piece of evidence. They point out that the commercial product you buy isn't pure HMTBA. And that raises a really fair question. Could this be its fatal flaw? Let's take a closer look. As you can see from this chart, only about 65% of HMTBA is in that simple, ready-to-absorb form called a monomer. The rest is made up of these bigger chains called dimers and trimers. So on the surface, this looks like a huge weakness, doesn't it? It seems like a big chunk of the product isn't, well, immediately useful. But here's where the story takes this incredible turn. Researchers looked into it, and what they found, as this quote says, is that those other forms are not a limiting factor. So why? It's because of the hydrolytic capacity of intestinal mucosa. Okay, in plain English, the animal's own body has the solution built right in. It turns out the gut is kind of like a brilliant chef's assistant. As the HMTBA passes through, the animal's own enzymes get to work and just chop up those bigger dimer and trimer chains into the simple, usable monomers. And they do it right on the spot, exactly where they're needed. So what looked like a critical weakness is actually no big deal at all. This particular clue was a total red herring. So after examining all the clues, debunking the false leads, and tracing the different paths, it is time to deliver the verdict in the great methionine mystery. And you know what? The final truth is way more interesting than just picking a winner. So let's recap the case, right? We've got different molecules taking different paths, being measured by decades of experiments that were often flawed from the start. The conflict was never really in the chemistry of the molecules. It was in the methodology used to study them. To get a real answer, you have to use tools that can see both the VIP taxis and the open gates. And believe me, this isn't just some academic debate. Understanding this has huge, real-world consequences. So, which one is better? The real answer is, it totally depends on the situation. For example, think about an animal that's under heat stress. Its body is trying to conserve every single bit of energy it can. So in that scenario, which do you think is better? The supplement that costs energy to absorb, or the one that's basically free? HMTBA's energy-free open gate becomes a massive advantage, precisely when that energy-guzzling taxi service for DL Met might be struggling. Context, it turns out, is everything. And that really leaves us with one last thought. This entire mystery was solved not by discovering some new molecule, but by realizing we were just using the wrong ruler the whole time. So the next time you see one of those fierce scientific controversies playing out, ask yourself, are they really arguing about the facts, or are they just arguing about their tools? Thanks for joining me on this investigation. Stay curious.